Hello and welcome to my presentation for the Scholarship Challenge. My name is Lizzie Bushnell and my topic is vector therapy for cancer elimination. It took a lot of careful consideration for me to reach this topic. For that, I would like to thank Dr. Stephen Palter, whose presentation on the past and future of medicine showed me not only where medicine has gone, but where it's going in the future. He also showed me that medicine and progress are invariably linked to technology. So therefore, advancements in medicine must be met with advancements in technology. I would also like to thank Dr. Craig Venter, who took computer programming, which is a passion of mine, and he also linked it with medical research, which is my career field of choice. He showed me that one can reprogram viruses and bacteria to suit one's purpose. And thus I arrived at my theory of using a vector to eliminate cancer. My theory involves applying existing gene therapy technologies to the eradication of cancer. Currently, these technologies are only being used to weaken drug-resistant cancers to make treatments more effective. However, I believe a vector that seeks out cells with a mutation in the p53 or the MMAC1 genes, the ones that are responsible for cancer, can be developed. This vector would in turn detect, infect, and destroy cancer cells with little to no damage to surrounding tissues. This is a multifaceted benefit, seeing as this could also be applied to terminal or complex cancers. In addition, it will not place the body under any unneeded stress, and once it has eradicated all the cancer, it will stop reproducing and die off. I feel that this is the future to treating cancer. This may seem easier said than done, but I've already thought of the implementation. The implementation should occur in 4 steps. The first phase would be to develop the vector. The vector must reproduce rapidly to keep up with the uncontrollably multiplying cancer cells. Also, it must have a low mutation rate, so it does not mutate into anything that may actually harm the patient. Finally, it must not trigger an autoimmune response in order to avoid putting the patient's body under any undue stress. Once it's been developed, then we move to the testing phases. The first phase of testing would occur on samples of cancers that are difficult to treat, such as late-stage, terminal cancers, inoperable cancers, and those with low cure rates or extremely aggressive cancers. In the Petri dish testings on these samples, the bacterial vector should multiply very rapidly and then cease once all cancer cells have been eradicated. It should then die off. You would see a very immediate effect upon the cancer cells. We would also need to test for possibly the cancer developing immunity to these bacterial cells once they've been introduced. In that case, we may actually need to combine it with traditional gene therapy for cancers in avoiding that drug resistance. The second stage, I believe, would be testing it on mice with cancer. I believe mice would be the ideal test subject because not only have we sequenced their entire genome, we have a lot of experience with testing mice with cancer. Furthermore, mice are also mammals, which makes them a comparable subject to humans. And they react similarly to substances the way humans do. So I believe that this would be the next step. I mean, you obviously wouldn't want to test it on a human right out the bat. That would be wrong. I believe the final stage would be small-scale clinical trials on volunteers with late-stage or terminal cancers. If it succeeds in treating them, it may actually save lives. I think that if it was as effective in humans as it would be in mice, then we would have a real cure for cancer here. It is possible. In conclusion, to sum up my main points, my theory is of bacterial vectors, and they are beneficial because they would cause no harm to actual healthy tissues. They are minimally invasive, and they would not cause any stress upon the body, as well as they reproduce inside the body, so it would make it a cost-effective treatment as well, and it would be effective in cancers that we have trouble treating today, such as aggressive, drug-resistant, late-stage, or complex cancer. As you can see, I do believe that the next stage of cancer fighting should occur on the cellular level, and if scientists get together behind this, progress is possible. We may live in the world where there is no cancer by the end of my lifetime, and that would be a fantastic goal for medicine. And with that, I leave you to consider my submission. Goodbye.